Take it away.
Cool. Great. So for our semester project, we wanted to create a computational diagnostic test for food allergy risk. Why food allergies? Mostly because the prevalence of food allergies have increased significantly over the last 20 years. The best way of measuring food allergy today is called the oral food challenge, which gives patients a high risk for going into anaphylactic shock if they're actually allergic to something. This is bad. We want to avoid this. So we are leveraging recent studies that have shown that epigenetic markers are correlated with food allergy risk by building a classifier to predict risk. The data that we're using is taken from bulk blood cell data. The problem we found with this data was that so many cell types together confound cell-specific methylation signals. So a goal that we'll talk about is how can we get cell type specific data from our bulk data? The three aims. The first aim is to use a method called deconvolution, which is just going from like bulk to cell type specific. The second is showing that using the cell type specific data can improve performance of a classifier. And the third thing we'll do is talk about comparing deconvolved data using a naive-based classifier with a more state-of-the-art gradient-boosted tree classifier.
So as Taylor mentioned, most of the data sets that were available to us use bulk methylation data, and this covers up a lot of the cell-specific signals that we want to uncover. Luckily, we can use a technique called deconvolution to estimate those cell-specific effects. The idea is that if we have reference methylation values for the cell types we're interested in, then we can estimate the cell fractions that are present in a sample. So here in this figure on the bottom, we've shown the two data sets that we use in this work. On the left, which we're calling Martino 2015, the data set is supposed to only contain peripheral mononuclear blood cells, which are the four on the left. But deconvolution actually detects a small fraction of the three cell types on the right as well. In a follow-up study called Martino 2018, the authors actually filtered their data so that it only contained CD4 T cells, but deconvolution again detected a small fraction of CD8 T cells as well. And as we'll see later, including these cell types in our analysis actually improves performance. So once we know the cell fractions of our bulk data, we can use linear regression techniques similar to those that we learned about for GWAS in order to estimate these cell-specific and phenotype-specific contributions towards the bulk methylation values that we observe. So we run this linear regression for every cell type, for every CPG location. If we do this analysis on only bulk data, we find that none of the CPG locations have statistically significant differential methylation. However, if we do deconvolution and use the 7 cell types from the previous slide, we find 90 CPGs that have genome-wide significance across all 7 of our cell types. So we want to take this a step further and use these cell-specific DMCs as features for predictive models. We found that the results from linear regression can be used really easily in a naive-based framework to classify patients as either control or disease. So in this figure on the right, what we're doing is comparing the performance of our classifier as we include more cell types in our analysis. So for the Martino 2015 data set, we show the performance of a naive-based classifier trained on bulk data only. That's the blue curve on the bottom. You can see that at 100% recall, it has only 50% precision. So it's essentially a useless classifier. If we include the PBMC cell types, which is the red curve, then we boost performance slightly. And then if we include all 7 cell types that were inferred by deconvolution, we get the curve in purple, which has significantly better precision compared to the bulk classifier. Similarly, for the 2018 data set, which supposedly only contains CD4 cells, our bulk classifier has even better performance. And then if we account for that small fraction of CD8 cells that we detected, we get the performance shown in green, which is the best of all our models.
A major question we had was, do you even need convolution? Can you just use a super powerful classifier like gradient-boosted trees? And the reason we chose gradient boosting trees, one is because they are more powerful than, of course, an IGBS classifier, but also they're very explainable, meaning you can extract which features or which like CPG islands most correlate to a prediction, basically. We ran a gradient boosting tree algorithm on 2 data sets, both data sets. One was 2015, which is shown as on the right, the green dot. This is like average precision recall over 10 runs. And then on the blue dot is 2018 data, which to remind us is supposedly just CD4 cells, and performance also increases. Main takeaway is having fewer confounding cell types leads to better performance, at least in these data sets. One question you might ask is like, why can't we use like deconvolution as well as gradient boosting trees? And the main reason, at least in the way that we've done convolution, is we get cell fractions for each individual, but not feature vectors for each individual, like methylation patterns that are cell type specific. We can go into more detail during question answer. Main takeaways are that deconvolution, or one thing I'll mention too is we need feature vectors that are like cell type specific to input into gradient boosted trees, but we can't get those from deconvolution. So the main takeaways is deconvolution does improve performance, but we're limited in the number of other kinds of models we can use. We theorized that if there was some way to get individual specific cell type specific data shown on the bottom here, then we could input that into a gradient boosting tree and improve performance even more. Special thanks to Lei and Professor Pellis' lab who helped us think more and understand deconvolution, and we'll now take questions.
Awesome. So usually it's better to go back to your summary slide every time you're waiting for questions. This way, it's not the first one, but the last one. So help people ask questions. Yeah. So one of the questions that I have is for the cell type specific. So basically, you mentioned that you only get cell fractions, but not cell type specific vectors. Whereas this diagram made me think initially that the convolution would not only give you the fractions, but also the vectors of activity for every CPG in every cell type. Can you tell me more?
Yeah, so go back to the
basically, otherwise it's a cell type fraction predictor, not a cell type.
Yeah, so in this case, the deconvolution methods that we based ours off of give you a cell fraction, and then they estimate cell specific effects for the control and disease phenotypes, but not for each person in the data set. And so that distinction is what allows us to implement an naive-based classifier, because we don't need class conditional distributions, but prevents us from using.
But can you feed that data? Can you feed the sort of the case versus control specific methylation effects to the GBM, the other classifier?
Not in a way that we were able to think of, but it's possible, and we would definitely be open to if there is.
The other question that I have is there's a lot of methylation probes out there. How do you deal with multiple hypothesis testing?
Yeah, so I believe we used a Bonferroni correction.
Okay. So did you also try shuffling your data between cases and controls?
You mean like shuffling or triangulating test sets?
You could shuffle individuals based on the label or if they're a case or a control, and then see whether the system is still able to find CPGs that predict a random assortment of cases versus controls.
I'm sorry, I'm not sure I totally follow.
So suppose that half the class on the right is cases and half the class on the left is actual controls. Okay. Instead of splitting it this way, I'm going to split it that way.
Assuming that some.
So there should not be any CPGs that distinguish a randomly shuffled data set.
Oh, I see what you're saying. Yeah, I mean, in the case where we use only bulk data, we essentially found no significant CPGs. And I think if we did random shuffling, we would see the same.
That would be worth testing. Yeah, because it's always reassuring to say, listen, we shuffle it, there's nothing. Yeah. So it's not that, hey, there's so many CPGs out there that any random partition of the data, I'll find some that are predictive. Because make sense?
Yeah, yeah.
Awesome. Great. Thanks. Great work.